All right, welcome everyone. So today we're going to be talking about networks, network inference, network analysis, and network applications. So we are entering a new module. So we basically talked about expression and genomics in the first genomes, and then gene expression and epigenomics. We are now in the midst of network structure and network motifs. And then we're going to be talking about deep learning next time. And then we're going to enter the population genetics and disease genomics module pretty soon. So today's goals is to look at network algorithms. How do we interpret networks and some basic linear algebra techniques for networks. So what we're going to do today is first look at networks and graph foundations and algorithms. We're going to talk about different network types, social, biological, computational networks, and then some different types of networks, and then how to represent networks as graphs, and then just very basic breadth first search and depth first search. And then we're going to talk about emerging global and local network properties in the real world, including global properties such as small world, scale-free networks, clustering, motifs, similarity, dynamics, as well as local measures of centrality, such as degree centrality, between centrality, closeness centrality, and so on and so forth. Then we're going to dive into a quick review of linear algebra with eigenvectors and singular vector decomposition and low-rank approximations. And then we're going to talk about sparse PCAs and how we use PCA and sparse PCA to sort of reduce the dimensionality of high-dimensional data, such as the gene expression data that we saw earlier in the term, but also nonlinear embeddings for projecting these into lower dimensional data, such as t-SNEs. Then we're going to talk about network communities and modules and guild by association, as well as spectral clustering, a very powerful way of partitioning networks based on eigenvectors, eigenvalues, and Laplacians. And then we're going to talk about network diffusion kernels and deconvolution. So networks date back to when Euler wanted to take a walk in the morning and wanted to cross all 7 bridges of Königsberg, but he couldn't figure out a way how. And then he basically proved that there is simply no way to cross all bridges in a walk. And this was expanded with Hamiltonian cycles across sort of these regular Platonic graphs, and then trees in Euler's, in electric circuits by Kirchhoff, and then the problem of four-coloring a map with any kind of planar map can be four-colored using only 4 colors. And again, there's graph proofs surrounding that. And of course, we now know that networks are everywhere in the real world. We talk a lot about social networks and collaboration networks in different types of fields, commercial networks. For example, this is all biotechs in Boston in 1991. Biological networks, which we're going to focus most of the time on, but also transportation networks and computer networks. And the graph algorithms that we're going to be talking about today are in fact applicable pervasively across all of these different types of networks. If you look at social networks in particular, they are now some of the biggest, sources of traffic on the internet. So Facebook is dominating WhatsApp, YouTube, Messenger, and so on and so forth. And you can see that a large number of those are in fact social networks. So, in terms of the monthly active users, we're talking the billions of active users. So 2.6 billion users for Facebook, to 2 billion users for WhatsApp, and so on and so forth. So social networks are everywhere, and social network analysis is extremely important. It can reveal patterns, properties, important nodes, subnetworks, classify individuals, and so on and so forth. In biology, there's a huge diversity of network applications. So we can talk about regulatory networks involving transcription factors and their target genes. So this is usually when a transcription factor binds in a location of the genome and then turns on that gene. So these are exactly the networks that we talked about at the end of last lecture, when we can predict individual targets of and individual edges of a transcription factor. And these are usually directed and signed and weighted. So directed because a regulator controls a gene, but the gene is not controlling the regulator, so they're directed. They're weighted because the strength of the regulation can be dramatically different between, say, B targeting C and A targeting C with different strengths. And they can also be signed in a repressive or an activating edge. Metabolic networks are usually connecting metabolites with edges that are enzymes. And usually it's a different edge that's pointing in one direction versus another direction based on the specific enzymes that catalyze the reaction in one direction or another direction. If you ignore the specific identity of the enzymes, these are usually undirected networks or bidirected networks, and they're usually weighted based on the rate of the reaction. Signaling networks are kind of upstream of the transcription factor network. So basically when you have a signaling protein, you have a cascade of signals that enters the cell. And again, these are usually directed and unweighted because it's usually just a phosphorylation or conformational change of the protein, and the amount saturates very rapidly. So you don't worry about the weight that much. Protein-protein interaction networks in biology are undirected and unweighted. It's basically telling you whether a particular protein interacts with another protein, and that's a bidirectional, it's a symmetric relationship. And then there's also functional networks, which are basically co-expression networks. These are not physical. These are more abstract. It basically tells you that this gene is co-expressed with that gene, and that gene is co-expressed with that gene, and so on and so forth. Even though there might not be a physical reason why they're co-expressed. It could just, they could both be targeted by the same regulator, or they could be, some other kind of mechanism, indirect mechanism through which they're correlated, but perhaps through the environment. Maybe they both respond to the same signal or to signals that tend to co-occur. And again, these are undirected and weighted networks that basically where the weight represents the strength of that correlation. So let's see who's making so far. Awesome. So 64, 29, 700. Okay. So there's many challenges associated with networks. So basically on one hand, you want to identify the building blocks of the networks. And that's what a lot of the lecture was about on Tuesday this week. So we talked about how to identify the regulators, how to identify the regulatory motifs and to do motif discovery, and how to identify their target genes. We can also use networks to predict cellular activity. So for example, you might have a particular network connectivity pattern and want to predict the expression levels, or to predict the gene ontology, functional annotation terms of a particular gene. Or you might want to infer the network from functional data. We talked about these co-expression networks. You might want to infer the structure of a co-expression network or the structure of a regulatory network using gene expression for the regulators, for the targets, and so on and so forth. Or you might actually want to infer the function that is being computed by the biological network. You might want to say, well, if A and B are expressed at a particular level, then I can predict X as a function of their levels. And maybe that function is not just an additive function, maybe it could be some nonlinear function, and so on and so forth. And beyond inference of these networks, you might want to understand the structure of these networks. So you might want to infer degrees or network motifs or functional modules in those networks. So again, network science is pervasive in biology. And again, these are mostly physical networks that we're talking about, but beyond these real world networks where the edges actually represent some, physical relationship, you could basically have the networks simply be about co-expression, or they could be, the nodes themselves might actually represent biological variables or abstract mathematical variables. For example, you could actually use graphical probabilistic models where you're modeling the joint probability distribution of variables where every node is a variable using the graph. So you can have Bayesian networks, which are directed, or Markov random fields, which are undirected. And you could actually represent the fact that information flows through this network, specifically through the edges. So if you have a set of variables, you can express the dependencies between these variables as edges. and therefore the independencies as absence of edges. And in this particular case, you see that the information can flow between subnetwork 1 and subnetwork 3 through subnetwork 2. And that all of the nodes of subnetwork 1, if they're if subnetwork 2 is observed, become conditionally independent from subnetwork 3. Let me give you an example. So suppose that the higher the activity of this node, or the higher the probability of this node, the more I will believe that this node is also on. So observing the value of this node flows some information about the posterior probability of this node, which in turn influences the posterior probability of that node. By contrast, if I have fully observed the posterior probabilities of this S2, then there's no influence in any change in S1 that will influence S2, and therefore S3 will be conditionally independent from S1 given S2. Okay, everybody with me so far? Awesome. So let's talk about these network and graph foundations. So how do we even represent the networks? So a network, more concretely, is a graph, which is more abstract. And informally, there's a set of nodes which are joined by a set of edges. The nodes are objects. They're represented as circles or endpoints or data or variables. And the edges are basically links between them. They're the lines that were represented here. They're the arrows or the relationships between these nodes. So a graph is basically a set of vertices and a set of edges. And the vertices is usually some number denoted by n, and then the number of edges is a subset of the full V cross V, all pairs of vertices. And that number is sometimes denoted by m, and then the number of edges is usually much smaller than the fully connected network. So here's an undirected example, and here's a directed example of a graph. So again, the vertices are ABCD, and the edges are unordered, so they're just sets. Whereas in a directed example, the edges are actually ordered. So AC is very different from CA. So, AC exists in this network and CA doesn't exist in that network. Okay. So we can represent these networks as graphs. We can have weighted graphs where the weights are associated with every edge, and these are generally positive, except for signed networks where they're sometimes positive, sometimes negative. You can have multigraphs or pseudographs where there can be multiple edges between the same pairs of endpoints. You can have digraphs where these are directed graphs where the edges have actually direction, and simple graphs where there's no multiple edges and there's no self loops. Okay. So you can represent the network using a matrix notation. So you can represent the network as either a list of adjacencies. So you can basically say, as we saw before, AC, AB, BC, CB, this is an adjacency list. Or you can actually represent it as an adjacency matrix. And in a symmetric network, that adjacency matrix is symmetric. And the edges represent that from A to B and B to A, it's the same weight, 1.5, and so on and so forth. So an unweighted network is a binary adjacency matrix that basically tells you, is there an edge at all between pairs of nodes? Whereas a weighted network has a real valued matrix that basically tells you the weight of each of those. And we talk about the degree of every node as the number of edges that connect to it. So the degree in an unweighted network here would just be 1, the degree here would be 3, the degree here would be 3, and so on and so forth. But in a weighted network, the sum of the weights of a particular node is the degree of that node. So basically the degree of B is 1.5 plus 3.4 plus 2.1. And that's the BE edge. That should be 2.1 here. So there's a little typo on the slide. So basically 1.5, 3.4, 2.1 is the weight of B. And that's the degree of B. And in a directed network, you can have the in degree, which is the number of nodes, the edges pointing in, and the out degree, which is the number of edges pointing out of a particular node. So again, this is the adjacency list representation and the matrix representation. All right, so there's very basic algorithms on networks such as breadth first search and depth first search for finding shortest paths, for finding connected components. So with breadth first search, you basically start with a vertex, you list all of the neighbors at distance 1, then all the neighbors at distance 2, all the neighbors at distance 3, etc. And you define a frontier that you move through as you explore your network. With depth first search, it's the opposite. It's like exploring a maze. You basically start from the current vertex, and then you move to another one until you get stuck, and then you backtrack, and then you move to another one until you get stuck, and then you backtrack, and then you move to another one, and so on and so forth. Okay, so very different ways of exploring your graph. And algorithmically, for breadth first search, you start with the vertex, you expand all the neighbors, you define that frontier, and you move forward. And there's algorithms that you've probably learned in 600 and 604.6, where the only edges non-traversed by the breadth first search link vertices within the same or adjacent labels, and you can define properties of that breadth first search tree, which result directly from the particular way of exploring. And a corollary of that is that the breadth first tree results in shortest paths. So basically, as I sort of maintain that frontier, I know that if I explore D through X, it's because that is the shortest path, and there's no other shorter path that I can get to D from, S, for example. So the correctness analysis stems from the fact that if there was a shorter path, then you would have found it earlier in your search. So you can basically follow these pointers and then infer all of the shortest paths. So for the depth of search, again, you sort of explore just like a maze, and you backtrack, and you can basically have, just a node weight that basically tells you when did I observe each of these nodes, and then color them as soon as you've actually observed one of them. And then you basically have these cross edges and these back edges as you're exploring your tree using this depth first approach that basically tells you if something is at a lower level or not. So you can classify every edge based on, whether it's a tree edge, which is basically part of your depth first search tree as you're exploring all of the nodes, or a back edge that basically leads from a node to someone that I've previously visited as an ancestor to the current exploration, or a cross edge, which basically points to someone who's not an ancestor, but who is already explored through another part of the tree. And then a forward edge is when you would actually find a descendant of that tree that you have already explored. Again, this is not a shortest path, unlike the breadth first search, and you might actually find a longer path to get to somewhere because you're going depth first on this. And then depth first search is a very nice way of finding connected components, for example, to basically infer if there's a path between them or an equivalence relation between them. So if a vertex has already been reached, you don't need to search from it. So you basically have a set of nodes that you maintain that have already been visited, and a set that have not already been visited, and you sort of only explore for the ones that are not visited. So this is just very basics of networks. This should hopefully be just a review for most of you. We can now talk a little bit about another topic, which is very common in networks, which are what are these emergent global and local properties of these networks. So there's a lot of real world networks out there. There's basically infrastructure networks, there's social networks, there's information networks, and of course, biological networks. So the infrastructure networks could be the internet, the power grid, transport networks, distribution networks. In social, you could have friends, you could have actors and what films they starred in, you can have co-authors of papers, you can have affiliations of institutions. For information networks, you have web pages, you have citations of papers, you have patents and file sharing and shopping lists and documents versus keywords. And in biology, we talked about physical networks, metabolic networks, regulatory networks, neuronal networks, and also ecological networks. So all of these networks, despite being in such different domains, have some emergent properties. One of them is the small world property. So there was this famous experiment in the 60s of Milgram, who basically coined the term of 6 degrees of separation, where any pair of vertices were in fact connected by short paths. Even though there are 6 billion people on the planet, you can send a letter by giving it to one person and saying, don't use the post office, only give it to someone you know, knowing that it's trying to reach this person in China. And what's really remarkable about that experiment is not only that there were only 6 paths, 6 hops for any one person to connect to any other person on the planet, but that people were actually pretty good about finding these paths with no global information. So every person basically said, well, you know, I obviously don't know anyone in China, but I know this person who travels a lot, so I'm going to give it to that person. And that person would basically say, well, yeah, I haven't traveled to China myself, but I know this other person who travels a lot to China. And then very rapidly, it would sort of make these very long paths by only sort of person-to-person transmission. Another property that became very famous a little bit later is this whole scale-free and power law degree distribution, this 80-20 rule, that 80% of the connections are actually made by 20% of the vertices. And this whole concept that there's very few heavily connected hubs, and that most of the nodes lie in the fridges, whereas the hubs are basically, you know, sort of responsible for most of the connectivity. And related to the scale-free, power law distribution, the model that Albert and Barabasi came up with to explain the scale-free property was one of preferential attachment. A rich get richer, which basically will lead to these power law distributions. The idea is that if I'm a random node and I'm trying to connect to someone, if I connect preferentially to someone who's very heavily connected already, that will eventually lead to these networks that are power law distributed. Well, you can also define this clustering coefficient, which is the average probability that the neighbors of a node V are also connected to each other. And this basically measures the density of closed triangles versus open triangles. And more generally, it's a measure of the frequency of all network motifs, i.e. the over or under representation of all of the subgraphs of size 3, 4, 5 is very frequently a consequence of this clustering coefficient. So these network motifs that I mentioned in the previous slide are basically thought of as building blocks of networks, i.e. the smallest meaningful units of these networks, and also somewhat interpretable circuit components. You can think of feed-forward loops and feedback loops and cross-regulatory loops and amplification loops. And you can discover those based on their over-representation compared to a random network. You could basically say in a randomly connected network, I might expect, you know, a small number of these motifs, but in fact, when I look at a real-world network, I see that these are very heavily overrepresented, and you can sort of measure that computationally using some kind of statistical measure. So, this is an example from one of our papers on the Drosophila mod encode project. That's basically looking at different types of cross-regulating edges where two Fs are regulating each other, two transcription factors are regulating each other and both regulating a common microRNA. And then here's some examples of ABC for each of those motifs. Here's another example of cross-regulating clique of TFs where all of them are controlling each other. Here's a feed-forward loop where, A can repress C, or, B can go through A, and so on and so forth. You can have a double feed-forward loop where A controls C through B or directly, and A also controls B through C or directly. And then there were, biological examples for each of those. You can also study the hierarchical organization of these networks. And this is an example from the same paper where we could basically see that there was this concept of a set of master regulators, which are actually very well studied, that were controlling a subset of sort of higher order regulators with them, and then progressively lower and lower and lower regulators below that, each of whom has mostly targets down from them, and then a set of microRNAs which were modulating that network and also enabling some nodes to some edges to actually point up the nodes of the trees. So there's a concept of hierarchical organization of these master regulators versus these local regulators, of degree distributions with in hubs and out hubs. In hubs gather a lot of information and then might sort of propagate it locally. And then out hubs are basically master controllers of a lot of other nodes. You can talk about the diameter of the network, basically how far does information travel through the network, perhaps in a breadth first search. And then you can talk about the modularity or the locality of these networks. Basically, can I sort of break up that network into modules, but also clustering the network into sub-networks. And also the direction of the flow, whether it's downward through these master regulators or back upward through these microRNAs. So there's a lot of properties that we can compute for each of those nodes. One of them is the centrality. And you've probably all heard about hubs in the network. And you can measure the centrality of a node as the degree centrality, which is typically the original way in which these hubs were defined, based on their number of in edges or the number of out edges. Basically, the number of neighbors can be used as a measure of importance or authority of a particular node. Eigenvector centrality is the sum of the centrality of all of the neighbors of a node, where it's high when V has many neighbors that are central themselves. There's cat centrality, which balances the number of neighbors and the neighbor centrality using a weighting parameter. There's the page rank, which dilutes the centrality of the flow out of a vertex by the number of neighbors. And this is actually what's used in Google search results. There's closeness centrality, which is basically what is the mean distance to other vertices from a particular node. And the betweenness centrality, which is the number of shortest paths that go through a particular node. And there's also the flow betweenness, which is the amount of flow through V, not just the number of paths, but in a weighted network, what is the total amount of flow, as if you're looking at electricity or a river or any kind of metabolic flow or any kind of distribution network flow. And there's also the random walk betweenness, which is a measure of diffusion. You can basically have some kind of sink, some kind of source, and basically you could ask how much of that flow goes through a particular vertex V. So we can actually have specific information about node pairs, about the similarity or the closeness. You could look at assortative mixing, at node similarity or node equivalence, and at the clustering of particular properties in cliques or k-plexes or k-cores. And you can also define graph neighborhoods with particular components. You can have a component which is any two nodes linked by at least one path. That's when they belong in the same component, or a k component if there's at least k vertex independent paths between two nodes. So basically, I get to go from A to B through k different paths, none of which share any intermediate nodes. So, I encourage you to read a lot more about these to sort of, dive into the properties of these networks. And there's several very nice books about those that I really encourage you to learn more about. So beyond basically components and k-components, you can basically think about these giant connected components of these networks where everything is ultimately connected with everyone. So you don't want to just simply say, oh, great, there's one component here. You can basically look for local community structure, locally densely connected components. So, there might be substructure within a particular cluster of friends. So we can talk about graph partitioning algorithms that can break into k clusters. And the simplest form of that is a graph bisection, which is basically saying, can I find an optimal path, an optimal cut that sort of maximizes the separation? And, this is usually an NP-complete problem, because the exhaustive search is basically 2 to the n, divided by square root of n. And there's mostly heuristics, basically. Or you could divide randomly and then reassign the members, and that's sort of a Kernighan-Lin method. Or you could use spectral partitioning, which uses a graph Laplacian that measures some type of diffusion. And we're going to talk a little bit more about the Laplacian later in this lecture. There's also a lot of community detection algorithms for discovering these coherent small groups and for maximizing modularity. Again, you could use spectral methods or betweenness-based methods and other types of methods. You can also use dynamic processes on those networks, for example, percolation or network resilience. You can basically either uniformly or non-uniformly remove individual vertices or individual edges or individual hubs, and then see, what happens to the connectivity of the network. If there's a network router failure, or if there's an attack on the network, or for example, if you vaccinate individuals during, I don't know, a hypothetical pandemic, you would basically like to know, how do I prioritize who I should vaccinate in order to maximize the resilience of the network, or to minimize the speed of spread in the community, and so on and so forth. There's a lot of models that look at epidemics on these networks for the spread of diseases, for susceptible, infected, recovered individuals, and that has been, again, all over the news. But you can now start asking, well, what is the R naught at the start of a spread, and what is the RT at particular times during that spread, and how fast are hubs infected in a network, and how is the rate of growth, for example, affected at different points on this network based on how fast are hubs versus other types of nodes affected in a sort of disease spread model. And these can be time-dependent properties of the disease spreading. Again, we've heard a lot about R naught, but the number that most people are using is RT, which is the number of individuals that a particular person infects at a given time point T. You can have dynamical systems of these networks to basically look at the rate of a particular property change over time. So that's where a lot of metabolic modeling happens in the metabolic network that we saw, with steady state analysis and fixed points. If you know the expression of particular enzymes, you can basically infer what is the steady state abundance of different metabolites in my metabolic network using this information. But you can also look at information flow through the network. Basically, if you're spreading messages, or what is the stability and the synchronization of these messages as they're spreading through the network. And you can also look at network search. Basically, you can have a distributed database and message passing, and also search through such a network. So who feels that they've learned some stuff today? So let's see. How are you guys feeling in terms of the, number of properties that you've kind of heard through? I mean, I try to explain each of them simply, but my goal is mostly to give you a vocabulary with which you can go and explore that field and learn more about. Awesome. So we have 25, 63, 13, 00. Okay, so now let's go back to centrality, this whole hubness of a particular node, and look at different metrics of centrality and how they can be shaped by the shape of the network. So the question is, how important is a particular node or a particular edge to the structural characteristics of a system? And the question is, well, great, I can use degree by itself. This is a very high connectivity node, so it's probably very important. But look at all of those high connectivity nodes. Are they all super important to the network? What if this particular node has relatively lower degree, but there's a lot of information that goes through that node to get to these remote nodes here? Maybe this node should be thought of as more important than that node. So we need different measures of hubness, different measures of importance for these different nodes. So as I mentioned earlier, the degree centrality is simply measuring the number of edges that are incident on a particular node. That's in an undirected, unweighted graph. If you have a weighted graph, then you can have the in degree versus the out degree. And if you have a weighted graph, then it's the sum of weights of all of the nodes incident. And again, in the directed graph, either incident in or out of a particular node. So you can basically measure alternatively, beyond the degree centrality, you can measure the betweenness centrality, which is asking how many shortest paths in the graph pass through the node that I care about, and then divide that by the total number of shortest paths. So we talked a little bit about shortest paths earlier, and you can basically say, well, what is the total number of shortest paths between I and j? and how many of those shortest paths go through node K, as long as I, J, and K are distinct. So if you have nodes with high betweenness centrality, they might actually be important in controlling the information flow in a network, and they might be worth preserving in resilience to sort of worry about an attack against that network. And the edge betweenness is defined very similarly based on the number of shortest paths that go through a particular edge, not just a particular node, divided by the total number of shortest paths. So here's an example. If I have, for example, node B, if I care about the betweenness centrality of node B, I can basically look at all of the shortest paths going from A to C, D, E that go through B, and from C to A, D, E that go through B, and so on and so forth. And you can see that out of all of the shortest paths that I see here, the ones that are underlined are in fact going through node B, and that's 4 out of 10, or actually 5 out of 10. So that gives you between the centrality of node B of 5 out of 10. Okay. The closeness centrality is basically the normalized inverse of the sum of topological distances in the graph. So you can basically ask just how close is every node to every other node. So you can basically look at what is the topological distance between every node I and, between node I and every other node J. So basically, what is the distance between B and every other node? And then the inverse of that is the closeness centrality. So you basically have node B, which is the most central in spreading information from it to the other nodes in the network. And therefore, if I'd like to broadcast a message by shortest paths, I might want to start with B to reach everyone. And then you can see the, between the centrality of, the closeness centrality of B is the highest, and then followed by A, C, and then further D and E, just because of that connectivity. So A and C are connected here, and therefore A is actually closer to all of the other nodes, whereas E and D are not connected, and therefore E and D are actually further from all the other nodes. And again, depending on the network, node B might appear more, betweenness central and more closeness central, because it's both closer to all of these other nodes and, in between all these paths, whereas node A actually has a higher degree of degree 5, whereas node B only has a degree 3. So that's, the most central node or the most important node definitions might actually differ based on what metric you use. So eigenvector centrality is extending this concept of degree by making the score proportional to the average of the centralities of the neighbors. So if you have basically, all of the neighbors of a particular node, then you can basically say what is the centrality of each of those neighbors, J, and what is the spread between I and J in my network. And notice here this Aij notation. This is the adjacency matrix notation, which is also a way to get the flow out of that node. So if I multiply a vector with a matrix, I'm basically getting the impact of transferring weight from all of these nodes to all of the descendants through the network. So this matrix notation of networks is actually a very powerful one. And again, lambda is some kind of constant. So in matrix vector notation, this is simply AX and 1 over lambda with a scaling factor. And to make the centralities non-negative, you can select the eigenvector corresponding to the principal eigenvalue, which is this Perron-Frobenius theorem. All right, so that takes us to this linear algebra representation of graphs and networks that allows us to do all kinds of more powerful things. So we saw just very basic properties of networks, very basic algorithms such as breadth first search and depth first search, and then some very basic emergent properties of networks, both global and local, and some measures of centrality. So now let's dive into the linear algebra view of these networks. So again, you can have a matrix interpretation of graphs where the graph, VE, can be shown as a matrix, where you can choose an ordering of the vertices, you can number them sequentially, and then fill in a V by V matrix that basically tells you what is the weight between every pair of vertices. So if I have vertices 1, 2, 3, 4, 5, and then weight A between A and B, between 1 and 2, I have basically 1 to 2 here. But then notice 2 to 1 has no weight. So this is not a symmetric matrix because it's a directed graph rather than an undirected graph. So again, that's the adjacency matrix or the incidence matrix of the graph. And the diagonal entries are basically the weights of self loops. So basically G has a self loop here. Sorry, node 5 has a self loop with weight G. And then if it's symmetric matrix, that's an undirected graph. And if it's a lower triangular matrix, then there's no edges from the lower number nodes to the higher number nodes. And if it's a dense matrix, then you can have cliques, i.e. edges between every pair of nodes, and so on and so forth. So now you can basically think about the information flow through that graph by multiplying your matrix A with a set of weights. So basically, if I have a set of weights X, which are, for example, the starting weights of my network, then I can ask, if I flow these starting weights through the network according to the weights of each of these edges, then what will be the final set of weights? So that's Y, which is basically the operation of multiplying that vector by that matrix. So the graph interpretation is that every node I has two values, labels xi and yi, and every node I updates its label to y using the x value from each of the neighbors scaled by the label on edge ij. So basically from a graph perspective, this is, a matrix operation that just flows information through that graph. So one way to think about this matrix operation is a transformation of the space of, this 5-dimensional space of weights. So the geometric interpretation of these matrix operations is that I am shearing my space of, say, a 2-dimensional space here by transforming every vector on that space by the matrix M. And some vectors, for example, the red one here, will not be affected, and other vectors will change their direction as the whole space is getting sheared. So I'm starting with a 2-dimensional space, and I'm ending with a 2-dimensional space, which is a transformation of the original space. Okay? So who's been thinking so far about these sort of linear algebra view of these networks and the sort of matrix operations of applying a set of transformations to an initial set of weights on each of these edges, each of these nodes? Okay, lovely. So 43, 36, 14, 70. So you can basically shear the space using a combination of rotation and scaling, or you could actually transform this operation into first a rotation, then a scaling along these axes, and then another rotation. So this is basically the basis of this, decomposition of these matrices. So I can think of my graph as a matrix operation, and I can think of that matrix operation as a transformation of the space, and I can think of the transformation of the space as decomposed into a rotation, a scaling, and a rotation. So the way to think about this is that if I apply my matrix transformation, which is my graph, on an input set of vector x, input set of weights vector x, then this is equivalent to first applying a rotation v, and then applying a scaling s, and then applying another rotation u. Okay, so basically m is the product of u, sigma, and v, where I first apply v star, then apply sigma, and then apply u because of the order of the multiplications. Okay, who's with me so far on this transformation of the space and how it can be decomposed into these parts, and how I can actually think of singular value decomposition and this type of transformation as to breaking them up into these components? Okay, lovely. So 50, 33, 880. So this is basically what eigenvectors and eigenvalues actually do to your space. So eigenvectors are basically those vectors for which the transformation by this matrix is equivalent to just scaling that vector by a scalar. So as you're thinking about the shearing and this transformation of the space, all of the points are going to be moving except for some vectors for which that direction of motion is in fact causing them to be collinear with their initial transformation, their initial direction in which they're pointing. So basically, the eigenvector of a matrix is simply a vector such that transforming that vector by that matrix is just a scalar multiplication of that vector. So if I have, for example, this transformation, which you can think of again as a graph of, from A to B and from A to A and A to B and B to A and B to B, that transformation of that network or that graph or that matrix on these weights or this vector or this sort of space in these two-dimensional, grid is effectively the same as multiplying that same vector by a scalar. So that basically means that, 1, 2 is an eigenvector because the transformation, and is an eigenvector of that matrix, because the transformation of 1, 2 by that matrix is equivalent to a scalar multiplication of that. So the question is, how many eigenvalues are there at most? So basically you're solving the system of linear equations of SV equals lambda V, which is basically nothing more than S minus lambda times the identity matrix V equals 0. And that only has a non-zero solution if, this is 0. And then this is an mth order equation in lambda, which can have at most m distinct solutions. And these are the roots of that characteristic polynomial, which can be complex, even though S might actually be real. So this matrix vector multiplication is basically the same as taking the eigenvectors and then transforming each of them by a scalar. So you can basically take this matrix vector multiplication as a way of, thinking about this, which is each of these vectors is a separate dimension onto which I'm applying a linear transformation. So in each eigenvector, S acts as a multiple of the identity matrix, but as a different multiple on each of these dimensions, on each of these vectors, on each of these projections. So any vector, say x, 2, 4, 6, can be viewed as a combination of the eigenvectors, which is basically, if I multiply this along, it's 2 times the first eigenvector, 4 times the second eigenvector, 6 times the third eigenvector. So thus a matrix vector multiplication, such as Sx, can be rewritten in terms of the eigenvalues and the eigenvectors acting on it. So if I think of, S on top of X, I can basically say, well, X can be simply expressed as 2v1, 4v2, and 6v3, multiplying through the eigenvectors to sort of construct that original matrix, that original vector X. So basically, the first dimension, the second dimension, the third dimension of X. And then that's nothing more than the corresponding product on the matrix with each of the vectors. But because these are the eigenvectors of that matrix, every such transformation of the eigenvectors is just a scalar multiplication of the eigenvectors, which basically gives me this linear equation here. So even though X is an arbitrary vector, the action of S on X is determined by the eigenvalues in the eigenvectors. And therefore, a cool observation is that the effect of quote unquote small eigenvalues, namely eigenvalues that have only, a small lambda, for example, is actually small. So it's a very nice way of decomposing the total strength of each of those. So let's see who's with me so far on all of this sort of linear algebra review, hopefully. That's lovely. Okay, so we are at 62.23.880. Okay, so basically for symmetric matrices, the eigenvectors for distinct eigenvalues are actually orthogonal to each other. So if I basically take the eigenvectors and the eigenvalues are not the same, then the dot product of these vectors is actually orthogonal to each other. And also all eigenvalues of a real symmetric matrix are actually real. And for complex lambda, if S minus lambda I is 0, and S is ST, that's a symmetric matrix, then the lambdas must be real. And all eigenvalues of a positive semi-definite matrix, which is defined by this, are in fact non-negative. So basically lambdas must be positive if this condition holds here. So the W transpose SW is greater than 0. So here are some examples of eigenvalues and eigenvectors. So basically, if this is my real symmetric matrix, then S minus lambda I is this. If I solve that for lambda, then there's two solutions. One of them is, 2 minus 1 squared minus 1 is 0. And the other one is 2 minus 1 is, actually, it should be 3 here. 2 minus 3 squared minus 1 is again 0. So there you go. The eigenvalues are 1 and 3, which are non-negative and real. And the eigenvectors are basically the corresponding eigenvalues for this. So I can just plug in lambda equals 1, and I can plug in lambda equals 3, and then solve for this. And the corresponding eigenvectors will be 1, 1, and 1 minus 1, which are basically pointing diagonally up and then diagonally to the left. So if you plug in these values and then solve for the eigenvectors, you get that. All right, so then the eigendiagonal decomposition is to basically carry out the same sort of transformation of this matrix S into 3 operations. And for square matrix with M linearly independent eigenvectors, namely a non-defective matrix, where the actual dimensionality of the matrix is the dimensionality that is proposed, rather than having some 0 eigenvalues, then there exists an eigendecomposition which has this property, where the columns of U are the eigenvectors of S, and the diagonal elements of lambda are in fact the eigenvalues of S, and all of the non-diagonal entries of lambda are actually 0. So you can basically think of the transformation S as basically the transformation of each of its independent eigenvectors, each scaled by the corresponding eigenvalue, and again transformed here as well. And this is unique for distinct eigenvalues, ordering these eigenvalues by the largest to the smallest. And if they're all distinct, then there's a unique decomposition here. So this is just a quick proof of why diagonal decomposition is possible. So if U has the eigenvectors as its columns, then SU can be written as S times each of these vectors, which is basically the same as multiplying by a lambda for each of those because of the eigenvector property. And then this is the same as these diagonal lambdas times all of the V's by sort of, expanding this out and then multiplying each of the columns by each of these rows, which basically gives you, again, the exact same thing as multiplying each vector by scalar. And therefore, SU is the same as U lambda, and SU U minus 1, so basically the inverse of U, is the same as U lambda, because SU is U lambda, times U minus 1, okay? And therefore, S is that decomposition, okay? So here's another example of diagonal decomposition. So you can basically have, this S with eigenvalues 1 and 3 and eigenvectors minus 1, 1, and 1, 1, which basically form this U. So if I invert U to U negative 1, this is basically the matrix with which I need to multiply that in order to get the identity matrix 1, 0, 0, 1. And that is, sort of the solution to that. And if I basically multiply all this across, then this becomes S. So if I have this 1 minus 1, 1, 1, which are these two, orthogonal vectors to each other, the corresponding eigenvalues, and then the inverse of U, that's basically the eigenvalue decomposition of S. So, and then, you can basically divide and multiply everything by the square root, and then you can get this orthogonal matrix where every column has unit length and is perpendicular to all the other columns. So you can basically transform this that way. All right, so that's symmetric eigendecomposition. So if it's a symmetric matrix, then there exists a unique eigendecomposition where this Q is orthogonal, and then Q negative 1 is the same as transpose of Q. And then the columns are basically normalized eigenvectors, and the columns are orthogonal, and everything is real. Singular value decomposition is very helpful when the matrix A is an m by n matrix of some rank R. And this doesn't need to be a complete matrix. It can actually have degenerate dimensions. Then there exists a factorization, i.e. a singular value decomposition, which is basically defined as follows, where for that matrix A, I can decompose it into an n by m matrix, U, an n by n diagonal matrix, sigma, where the diagonals are the eigenvalues, or the singular values, and then an n by n matrix. So the columns of U are orthogonal eigenvectors of not A, but AA transpose. And the reason for this AA transpose is that I want my dimensions of A to actually be matched. The columns of V are orthogonal eigenvectors of A transpose A. Again, this is an n by m matrix, and this is an n by n matrix, to basically take this not necessarily square matrix and then make it eigenvectorable. And then the eigenvalues of AA transpose are basically the eigenvalues of A transpose A, and these are nothing but the square roots of the basically the singular values are basically the square root of those eigenvalues. Okay, so basically the singular values sigma are basically the square roots of the eigenvalues of this A transpose A matrix. And the singular value decomposition is very helpful in basically being able to decompose matrices that are not necessarily square and not necessarily symmetric. So here's an example where you're basically looking at this matrix A that has, a 5 by 3 dimension. And there's, for example, 3 full eigenvalues here. And then the other 2 dimensions are, not quite matching. And then you can decompose that into a U and a V, which are 5 by 5 and 3 by 3 to match these 5 by 3. And then 3 eigenvalues, so singular values here, which are basically the square roots of the eigenvalues of AA transpose. And, conversely, you can have a, dimensionality in the opposite direction with the remaining ones here and then the remaining ones there. Okay. So singular value decomposition can be done for such a matrix here, where you can basically find the singular values of these 3 by 3 and these 2 by 2 vectors, sort of matrices. And then you can arrange the singular values in decreasing order of magnitude, with the most important to the least important. So the reason why singular value decomposition is so awesome for matrices and for graphs and for networks and for correlation matrices in general is that it provides a very natural low rank approximation. So singular value decomposition can be used to compute an optimal low rank approximation of any matrix and any graph. So this is an approximation problem where you're trying to find the kth order, the rank k approximation of a matrix A, as a way to minimize the distance between the original matrix and this low rank version of that original matrix, using this Euclidean norm or Frobenius norm, which is basically the square root of the sum of all of the square differences between A and X. Okay, so I'm basically taking the sum of the squares across all dimensions I and j, and then taking the square root of the sum of the squares, and that's what I'm trying to minimize. So the kth approximation, the kth rank approximation, and X are both m by n matrices, and we want k usually to be much, much smaller than the actual rank of the original matrix A. And then the singular value decomposition of that is amazingly trivial. It's basically you're taking the U sigma V transpose decomposition, and you're just keeping only the k first singular values. And if you just set all of the remaining r minus k singular values to 0, you effectively can prove that this is the specific matrix that actually minimizes this difference And, in column notation, this is basically the sum of rank 1 matrices, all of this. Okay. And then the error is basically the thing that minimizes that. And it's basically simply the k plus 1 eigen value of, singular value of the original matrix A. Okay. So the question is also, well, where do I stop? How far down the list should I go in zeroing out these singular values? And there's a very cool theorem about how to choose rank K by looking for non-random eigenvalues and eigenvectors. So the question is, what does the network, the matrix, look like for a random matrix? And does it have some non-random structure? And this Wigner semicircle law is basically telling you that the distribution of singular values of a random matrix form a semicircle. And this is the Tracy-Widom distribution, the probability distribution of the largest eigenvalue. And if you look for non-random eigenvalues, basically this tells you that, yes, the non-random eigenvalues are going to be outside that semicircle. So one way to choose your low-rank approximation is to basically see at what point do I get non-random eigenvalues, and at what point do I get all of these sort of semicircle distributed eigenvalues. Okay. All right, so let's see who's with me so far. So we've talked about eigenvectors, eigenvalues, and also singular value decomposition. We saw how this can be used for low rank approximation and this Wigner semicircle law for actually choosing the dimensions that I'm going to be zeroing out on my singular value decomposition. All right, so 45, 27, 18, 9, 0. All right, so this is, singular value decomposition with, we can now look at how we can make this even sparser. So basically, how do we carry out principal component analysis by basically decomposing my matrix in a way that sort of creates sparseness? And the reason for that is that if I carry out this principal component analysis, if I basically do a decomposition of my M arrays and N genes through my gene expression matrix, where I, for example, have 20,000 genes that are measured across 100 different conditions, my number of genes is much, much larger than the number of conditions. The question now is, what is this data telling me? And for every eigen gene, you can basically think of it as being expressed only in the corresponding eigen array with a corresponding eigen expression level. So I can basically take my M by N matrix of gene expression data, and I can decompose it into this M by M sort of matrix of arrays, of experiments. This is basically how experiments correlate with each other, basically. This is how genes correlate with each other. And then this is how the eigen combinations of experiments and genes. So what this looks like is, if I take, for example, data set like the National Institute of Standards, handwritten characters, and I do a PCA, a principal component analysis on that, then every one of those points is a 0 character decomposed and projected onto a 2-dimensional space, which corresponds to the principal components, the lower dimensional projection of the space. And you can see here just how beautifully this separates out the 1s all the way out there, the 0s all the way up here, the 8s and the 9s, for example, sort of are partly overlapping here. And then up there, I have the 7 and the 8, the 7 up here and the 9 and so on and so forth. So there's a lot of intuitive notion that comes with where are those characters in this 2-dimensional space. But you can see also some drawbacks of PCA, and these principal components are in fact linear combinations of all of your variables, all of your genes. So every principal component corresponds to a loading vector, which is the columns of V. And the loadings are coefficients corresponding to variables in the linear combination. And these can be difficult to interpret because you have actually the full combination of all of the dimensions for every one of them, no matter how infinitesimally small they might be. So what sparse PCA does is that it seeks to reduce the number of explicitly used variables, i.e. genes, by modifying principal component analysis so that the principal components have basically sparse loadings, so that I basically zero out a lot of the smaller values. So that's sparse PCA. And it basically writes principal component analysis as an optimization problem similar to a regression. And it uses this lasso approach of least absolute shrinkage and selection operator invented by Tipshiran in 96, where both the variable selection and the regularization are combined, and it produces basically these sparse models. So the result is a modified set of principal components with sparse loadings. So you don't have the original principal component, but you have a modified set that best capture the sparsity constraint. So the question is, how do you select this sparseness? So basically, this is the linear regression problem of expressing PCA using Lasso as sparse PCA, where you have basically a set of input variables, X, a set of output variables or response variables, which are a function of X plus some noise, and then a set of regression coefficients. And again, this is applicable not just in gene expression analysis, not just in networks, but also in genetics. We're going to be talking a lot about these beta coefficients when we start looking at heritability and when we start looking at genetics and sort of multiple variables combining together to sort of infer this, output response variable. So this is basically a multivariate linear model where you basically have a function of all of these input variables, which is a set of beta coefficients multiplied by the input variables. Okay, and we're trying to infer what is that set of betas. So the linear regression problem is basically given n observations and p predictors. The goal is to estimate these coefficients beta given some error epsilon. So you have a set of training data, you have a set of labels for that training data, and the labels are basically the output variables. And then given these labeled training data, where I kind of know the answers for many of them, the question is, what should be the coefficients that minimize this error? And the least square solutions for that is, given my training set of input vectors and output variables, to infer the residual sum of squares, this RSS of the betas, to basically optimize the betas that minimize this residual square error given some training set D. Okay, so in 2 dimensions, this is basically asking what are the squared errors, the deviation from the output variable Y that a linear regressor would infer versus the actual observed variable Y, and the deltas between the two are effectively these differences. So with sparse PCA, given a lasso penalty, you have your same training set D, where you have your input variables and your output variable. You have a residual sum of squares, which is basically, the difference between the beta regression of your input variables and the Y variable. And you have a lasso penalty, which is basically a linear cost of adding up all of the absolute values of all of your coefficients beta for some, lambda positive, for non-zero lambda. And your objective is to basically find a set of beta, not optimal betas as I had before, but now a set of beta lassos that minimize not just the residual squared errors, not just the residual sum of squared errors, but also this linear penalty on beta, this linear sum of the absolute values of your beta. So the advantage of this is that Lasso is continuously shrinking the coefficients towards 0 and produces a sparse model by basically minimizing the weights of these coefficients. And you can think of it as a variable selection method, where you're basically selecting which of your variables are the most important, which of your dimensions, which of your genes are the most important. The downside, of course, is that the number of selected variables is actually limited by the number of observations. And the microarray expression data has, many, many more genes than arrays. So it selects only one of the highly correlated variables, and it doesn't care which one is in the final model. So that's a bit of a problem, because you might say, well, hi, it only depends on this gene, but not on all these other genes, which are equally good predictors. So there's another way of regularization, another way of sort of, sparsifying this model, which is, instead of this lasso regression, which is this linear penalty on the weights of the coefficients, to basically use this ridge regression, this L2 norm, this quadratic penalty, where you're basically looking at the sum of the square weights. So the advantages of the L1 is that it's more robust, where the outliers don't matter as much. But the disadvantages of the linear regression is that it's less stable in the gradient ascent, and it has multiple solutions. The advantages of the L2 ridge regression is that there's always one solution, and it's a more stable gradient ascent, but it's less robust. Basically, the square differences of outliers have a much stronger effect. So, as I increase these weights, I basically have a stronger effect. So one way to sort of combine the lasso and the ridge penalty is to have a combination of weights between them, and just have, this L2 and this L1 each weighted by some kind of combination. And that's known as elastic net. So it basically combines the L1 and the L2 regression, and the limitation of lasso is removed by the ridge constraint, and all variables are actually included in the model. And then there's a grouping effect where you can select a group of highly correlated variables once one variable among them is selected, and then Lasso will select only one of them, and it doesn't care which one, whereas Ridge will allow you to include multiple. So what Sparse PCA does is that it constructs a regression framework in which the PCA can be reconstructed exactly, and it uses lasso, ridge, or elastic net regression to construct the modified principal components using these sparse loadings. So you can basically reconstruct PCA in this regression framework. The idea is that every PC is a linear combination of the variables, and its loadings can be recovered by regression on the P variables. And then you can solve PCA by regression by using this decomposition, this singular value decomposition of your X, given these principal components of X. And then suppose that your beta ridge, these are the modified betas that follow the ridge constraint, are basically the ridge estimate given by this minimization over beta of the differences, but also the lambda weights times the beta coefficients. So, and then this, beta ridge regression divided by the absolute value of the beta regression is basically this V hat that you can select for each of your vectors. So you can basically reconstruct this using the loadings of the PCs exactly by a linear regression problem. And this is not an alternative to PCA, as it uses the results of PCA, but then the ridge penalty does not penalize the coefficients, but it ensures the reconstruction of the PCs. And you can add the lasso penalty, the one-dimensional penalty, in addition to the two-dimensional penalty, the absolute values in addition to the squares, to penalize the absolute values of the coefficients. So you can basically reconstruct SPCA in this framework using this, there's residual sum of squared errors, this ridge regression, and this linear regression, and then select the ith principal components of each of your, eigenvectors using these sparse loadings, such that the ith component is this sparse principal component rather than the original principal components. So if you basically have, say 10 data points, 10 variables, you use this model to generate the data with 3 hidden factors, and then you basically have, this particular, residual error, you can basically say, well, I can have basically 4 variables which are associated with V1, and another 4 variables that are associated with V2, and another 2 variables that are associated with V3. Notice that I have a small number of weights because I'm reusing the same sets of weights. So rather than having a large number of parameters, I now have a small number of parameters, but these parameters are in fact capturing multiple variables jointly, as opposed to sort of only using one of the genes at a time. So Here's one example where you can basically simulate 10 observations, and then you can expect to derive 2 principal components with sparse loadings, one from 5 of these variables, one from another 4 of these variables. And basically, if you do this sparse PCA, what you see is that there's a single vector that captures the first 4 variables, another vector that captures the next 4 variables, and then that's it. So you only have 2 parameters, even though you're actually capturing all of your variables as inputs using the sparse PCA. All right, so that's all about linear decomposition, where you're basically selecting a linear set of projections for this, but you can also carry out nonlinear embeddings. So you can basically say, well, I have a very high dimensional data set, and I want to project it into a lower dimensional space by preserving the local neighborhoods. So here's one example where I've projected it, but now this red point that used to be close to all of these blue points is now far from all of these blue points. So what I would like to find is a lower dimensional projection that actually maintains all of the local neighborhoods, basically what are the closest points to the local proximities. So what this t-SNE method allows you to do is take this very high dimensional space and then measure the set of neighbors given some bandwidth such that the conditional of observing j given I, basically this proximity measure that is basically looking at this xi minus xj squared divided by some sort of geometric fall of minus, divided by this based on all of the other local points. You can basically set your bandwidth of how many points are you considering to basically make sure that your conditional has some kind of fixed, what is known as perplexity, which is sort of this neighborhood that you're looking at, which is the effective number of neighbors that you're looking at, such that you basically match the local proximity of the points that are near you in the original space back into this lower dimensional space. So you basically are choosing a nonlinear embedding, a nonlinear sort of lower dimensional projection that minimizes the divergence between your low dimension that you're projecting things into and your high dimension. So you can basically look at the similarity of the points in this high dimension, Pij, and then you look at the similarity of the points in this lower dimension, Qij, basically projecting every point xi into a point yi in this lower dimensional space, and then match the distance similarity functions appropriately between the two. And that's where this t-distribution comes in. So you're basically using a student t-distribution to avoid this overcrowding of having all of the points be on top of each other. So you can basically use a gradient method to find such an embedding in this lower dimensional space that minimizes this KL divergence of this lower dimensional distance versus the original higher dimensional distance. So with very large Pij, you can basically model it with a small Qij. That's basically a large penalty. But if it's a small Pij that's modeled by a large Qij, then that's a small penalty. So basically it's okay to separate nearby points, but it's not okay to bring distant points together. And then you can use a gradient function that looks at the difference of this cost function C on that KL divergence, which is basically taking the derivative of that with respect to the yi that you're sort of optimizing now, basically the new lower dimensional projections. And then this is, minimized according to that function of basically minimizing these distances. And then the original distance, the distance in the original space versus the distance in the transformed space, we're trying to minimize all that for all I different than j. So you can basically interpret these gradients as to a high dimensional distance, versus a lower dimensional distance, and sort of where are the corresponding weights, the corresponding penalties. And then this is the principal component analysis of these handwritten digits, where you can basically see how these are actually, spread across the space. The same thing now onto this lower dimensional embedding that maintains these local distances is in fact looking like this. So you now have digits 6, 5, 3, 8, 1, 2, 7, 4, 9, etc., each clustered with many other digits of the same type. And again, what this did is that instead of sort of having a linear projection across this sort of lower dimensional space, you now have a nonlinear embedding where you basically take this extremely multidimensional space of, say, 64 by 64 pixels for every one of these characters. And, this very, very, hundreds of dimensions space is now projected onto just a two-dimensional space that maintains those distances. So you can basically look at the effect of changing the perplexity. That's the number of neighboring points that you're looking at, whether you're looking at only 5 points, 30 points, or 50 points, and then how you're moving along these different recommended steps. And you can see that, you can, with various different levels of perplexity, maintain the original structure of your data. And then again, here, if you're looking at perplexity 5, you're looking at only 5 neighbors at a time. And that basically creates a lot more additional structure that is not visible if you're looking at larger numbers of neighbors, like 30 or 50. And then as you're going through these iterations at step 10, step 20, 60, 120, and 1,000, you basically are learning this two-dimensional embedding, this nonlinear lower dimensional embedding that is sort of capturing those distances optimally as you're sort of searching through that space. Again, the spread of the original points is not that meaningful. You basically can have, a lot of points together or very clumped together, and then it sort of is invariant to that. You still get the same type of clustering. And then the between cluster distance is not necessarily preserved as you're doing this. Basically, you can have the green be far away from the other ones or not. And you can also have some false clusters appearing if your perplexity is too small, because you're sort of looking at the local clusters that are forming, as opposed to having a high enough perplexity that captures the number of points. So this is basically the method of choice for a lot of the high dimensional data that we're using in single cell genomics. And we're going to come back and talk about those. So I'm just going to cover one more topic and sort of how do we discover these network communities and modules using this linear algebra technique that I was describing earlier, and specifically how we can in fact infer optimal cuts through the network using this spectral clustering approach. Hi, everyone. I lost my connection momentarily, so I apologize for that. So this, so basically what we're going to look at very briefly is spectral clustering, because it builds on all of these linear algebra infrastructure that we've looked at. So we've seen this adjacency matrix that's basically telling you who is adjacent to you in this space. There's another matrix which is very powerful, which is the Laplacian matrix, which is basically the degree matrix minus the adjacency matrix. So the degree matrix is basically a diagonal matrix that tells you the degree of every node. And then the adjacency matrix is, what we just saw. And the Laplacian matrix is basically looking at the number. So on the diagonal, you basically have the degree of every one of those rows, columns. And then on the off diagonal entries, you basically have the, minus the adjacency matrix. And notice here that every row sums to 0, because that's sort of, the definition of the number of edges minus those edges. So one way to think about the Laplacian, and one intuition about it, is that it gives you a set of gradient concentrations or a set of spring forces that are pushing you away from this, space of your matrix. So you basically have, some spring loadings here, and then the forces that are pushing you away from each of those. So what spectral clustering does on a network is that it learns the network modularity by calculating the Laplacian matrix, by taking the adjacency matrix and then subtracting it from the degree matrix, and then the eigenvectors of this Laplacian matrix. You basically eigen decompose this Laplacian matrix, and the eigenvectors of the Laplacian matrix are such that the first one is always 0. And the second one basically gives you a cut through the network with some of the nodes pointing in one direction and some of the nodes pointing in the other direction. And that allows you to find an optimal cut through this using this sort of spring and pulling intuition. And then, the lower dimensional vectors basically give you additional cuts, or you can simply repeat the procedure for each of those and sort of find these, partitions of your network. All right. So we covered a lot today, but I hope it gives you this introduction for how to deal with graphs and how to deal with networks of basically, how and everywhere you can find these graphs and some basically very basic graph algorithms, some emergent, basically global and local properties of these graphs in the real world with small world scale-free properties. And then this huge power of eigenvector and singular vector decomposition for low-rank approximations. And then how to formulate this sparsity constraint of not necessarily using all of your genes, but using only a subset of your genes and a subset of your dimensions, using this sparse PCA with this ridge regression and lasso regression and this combination of the two in this elastic net formulation. And then some nonlinear embeddings with t-SNE, and then also how to find communities using this spectral clustering of taking the Laplacian and the eigenvectors of that Laplacian matrix as a way to partition the networks. All right, so that's where we're going to end today. So the last question is, who feels that they've learned something helpful today? So let's see. Lovely. So we basically have 38 fives and 63 fours and then 0, 0, 0 afterwards. All right. So thank you, everyone. And then see you on Thursday for deep learning lecture. So bye, guys. Thank you.
Thank you.
Thanks. Thanks.